All right, so I guess we can get started. So I'm Claire Brown from the Advanced Bioimaging Facility at McGill University, and I'm also part of Canada Bioimaging and Bioimaging North America. And I would just remind people maybe to just introduce yourself with a short introduction like that if you ask any questions, just so we can get to know the other people in the community. So thank you for coming for our expansion microscopy group today. We're going to have a journal club style today, but we had a couple of more logistics questions beforehand. So one thing, so Natalie Wu is a master's student in my lab who's working on expansion microscopy and has really spearheaded this working group. And we were chatting a bit about the 1.5 hour time for the meeting and whether that might actually be prohibitive to some people, just sort of saying, I don't have enough time for that. And I think the last couple of times we've met, we've also finished early. So we wanted to just get to a quick poll to see if people think that 1 hour would actually be better or sufficient for this meeting. And of course, we always try to keep on time. So if we did choose 1 hour, we would keep it to 1 hour. So I'm going to ask you to just go to the reactions tab. And I think I don't see the yes, no. Oh, no, under the participants tab. I think it is. All right, I'm not finding it now. There's a yes, no poll, but I'm not seeing it under the participants tab. I don't know if it's our version of Zoom. Maybe we can just go with a thumbs up and I can take a screenshot. So if you agree that 1 hour is enough, you can put a thumbs up under the reactions tab. And if you don't agree, then don't put a thumbs up. And we can do a quick screenshot. So I count 10, including Natalie and I. And then 11. And we have 3 RMS people who I don't think will probably vote. So 11, we have 11 out of 18. So it's a little bit split. Is there anybody who would prefer the 1.5 hours who would want to voice their opinion on that? Anybody have a strong feeling that way, or was it more that you just don't have a strong opinion either way?
So there's a comment in the chat. Marina asked, could we have a place for online discussion on a Google Doc? And then 1 hour would be fine if we had that.
Okay. So the idea is that the meeting being 1 hour would be fine if we had a place to have discussions in between meetings. Okay. So that was actually the next item on my list. So great suggestion, Marina. Natalie, do you want to maybe present what you had been thinking about, and then we can go from there?
Yep, for sure. So pretty much like Marina said, we've been keeping with Google and the Google platform and Google Docs. So I was thinking that we can have a Google Doc essentially just for, like you said, an online discussion board where every time someone could ask a question, and then they can make their question as like instead of a normal text, you could put it as like a heading on Google Docs. And then that can create a side panel with like a table of contents listing out all of the questions. This format would mean that whoever asked the question would have to check back regularly to see if anybody has responded. And for those who will be responding to that question, if you could just either highlight and comment or like write beneath the question and list it out as like write your name or your contact, like email or something like that, and write your response on there. So you wouldn't be getting notifications per se every time somebody asks or comments, but this would be one way to kind of have like a sheet just allocated to people asking questions and responding. And so this was just an idea. It's open for anyone else if you have any suggestions on how we can stick to the Google platform, but also kind of have this discussion board format.
So we know there's lots of other resources like Slack and Discord and other places, but we find, at least so far, that the Google platform seems to be working quite well. It's just a matter that people would have to go and check once in a while to see if a question had been posted or if their question had been answered. So any comments on that? So I think Natalie already has a template started up. So what we could do is set that up and revisit it at the next meeting if we think it's working well. And if we think it's not working well, we could explore other options.
Yep, I can add that to our master document, and I'll include like an example on the template for how people can, or I'll write the instructions on the top to include like how people can ask a question and answer, and then a little example at the bottom. And I'll link that to the Google Doc that we already have.
That sounds great, Natalie. Thank you. All right, so we'll move into our main topic today. So just one comment: if people do have suggestions for speakers, or if they want to volunteer to present at any of our meetings, you can go to the master document, and there's a link there for all the different forms. I'm going to put that in the chat. And there's a link there for speaker suggestions or sign up sheet, and you can just link on that. It's under the Google submission forms, and you can suggest a speaker or volunteer yourself. Natalie did work pretty hard to find a speaker for today, and I guess busy time of year for everybody, and we were unable to find somebody. So we decided to do the journal club format, which I think is also great. And I think, that could be led by anybody in the group if you find a paper that you think will be of interest to the community. But it'd be good to have people speaking about their own work as well. So just a reminder to fill that out. And the other forms are there as well. If you have suggestions of fluorescent proteins, dyes, reagents that work well or don't work well, meeting topic suggestions, and we also want to continue to collect a list of publications and maybe make that into a curated list that we could post on the RMS website or elsewhere. So I'll turn it over to Natalie now. So Natalie has reviewed this article about a new expansion microscopy technique from the Boyden lab. And this is really a journal club. So Natalie's going to present the figures, but we really hope this will be a open discussion with the group of how people have interpreted the data, if people think the data is good quality, if there's anywhere we think things could be improved, or other areas that could be explored a bit more, then, feel free to do, we can do a critical review of the work. So I'll turn it over to you, Natalie, to go ahead and present.
Perfect. Thank you. So yeah, maybe could I just get a show of hands, like how many people here have actually, had the time to at least skim the paper, maybe take a look at it, so I know, how much of a summary kind of thing should we do, or if we can just dive kind of straight in. So if you've read the paper, could you, I guess, like raise your hand? Okay, that's pretty good. All right, thank you. So I'm just gonna share my screen here. Like Claire said, it's gonna be pretty much just diving into a lot of the figures and discussing that stuff. Yeah. Okay, so can everybody see my screen? Work. Okay. So that's for those of you who haven't had a chance to look at the paper. We're going to be discussing this new paper, like Claire said, on revealing nanostructures in brain tissue via protein decrowding by iterative expansion microscopy. And this is a very new paper. It was published September 2022 by the Boyden Lab. And this is kind of their extension of their iterative expansion microscopy, or a variant of it. And so I guess first thing is the aims of the paper. Anyone want to feel free to just kind of unmute yourself or raise your hand and kind of, I guess, let us know what you think, or type in the chat what you think the aims of the paper was and why this one is so different from all the other ones. I can give you a minute or two to do that before I kind of give you my take on it, I guess.
So I just put a link in the chat to the paper. If anybody doesn't have access, please let me know and I could send the PDF by email.
Okay, so I guess maybe you guys are all still typing, but for me, I think the main aim, yeah, I think that's Claire, if you don't mind sending the PDF. I don't know if there's
I'll send it in a second, Brian.
Okay.
I'll just comment that we avoided sharing the PDF by email just because I don't ever mind sharing one-on-one, but I think if we did a mass email of the paper, I don't want to get in trouble for copyright rules and so on. So I know we're looking at it with an educational lens today, so I think it's fine. But
yes, we'll give a minute or two for those who didn't have access to the link or to the PDF to also take a look before we give any spoilers, I guess.
Chris, could you put your email address in the chat?
Guess I'll give it about maybe 30 more seconds. So yeah, if anybody has any comments, feel free to jump in and let me know your thoughts. Otherwise, like Claire said in the chat, she said to obtain super resolution imaging with standard microscopes and ensure all proteins are labeled. So yeah, that's very true, and that's kind of the main thing for like expansion microscopy too, where you kind of want to kind of keep going to see, generally in expansion, you just want to physically expand it and make use of that property of the spatial, I guess, expansion. Whereas in this one, they're kind of combining iterative expansion microscopy. So normally in their previous paper, when they talked about just iterative, so IEXM, that technique doesn't retain the proteins. And so for this technique, they really want to achieve this like super resolution down to 20 nanometers while also being able to, so like using that. And they wanted to the first round of expansion would be to decrowd the protein structures before they label, and then combining it with this other round of expansion to really label it and get into the spaces that was originally not accessible. And so the methods that they use in this paper, so I kind of just mentioned a few briefly, and if you think of any, feel free to just kind of pop it in the chat. So like I said, they first used iterative expansion microscopy. They took that technique that they created not too long ago and really just added on to it. They use some validation techniques like DNA paint, which is known for super resolution imaging. And so like I said, the main technique that they used was just doing 2 rounds of expansion. The first round, they really focused on retaining the proteins of interest and anchoring all that to the hydrogel, or just creating this like extra this space for the molecules to be able to, the antibodies to bind afterwards. And then in their second round, which is where they were able to restain after and look at all these structures. And they used STORM as one of the validation techniques in addition to DNA paint. And they also did one other technique where they stained it before expansion and then restained it again after, so to achieve this within sample validation. And so I guess we can move on to kind of just the figures and like the results that they obtained. So this is where I would kind of like more people to participate, because I am fairly new in this field. So I would like you all to kind of give me some tips and pointers on what you guys think they did well or they could have done better in terms of presenting these figures or results. So the first one I included was pretty much just their graphical abstract, where they were just showing how you have a brain slice tissue. And then normally if you do the pre-expansion staining, so on the top, I don't know if you get some right. So on the top here in this figure, or this part of the figure, they're showing that, a lot of these structures that they're interested in the synapses are very clumped together. They're very crowded. Your antibodies can't really bind to it. And so at the end of the day, you're not really getting as much information as you can, because you just can't label everything. And so this is the EXR, so expansion revealing technique that they suggest in this paper, where you link it all to a hydrogel and then you expand that. And then that allows this added spatial factor allows for the antibodies to really attach to your molecules of interest and then to actually be able to see it after 20x expansion. And so anyone have any comments on this technique? Have they tried it or?
I do have a comment, actually. Brenda Jarvis from Vanderbilt. I'm new to the whole expansion world, but I've done a lot of work in 3D staining. And one of the issues that I have personally run into doing 3D staining is when you use SDS on any tissue, it tends to warp or destroy epitopes. So if you do the expansion first, when their process uses SDS, the first thing I thought was, isn't that going to destroy some of your epitopes?
Yeah.
So immediately you're going to wreck some of these epitopes. That's my first thought. So you.
Yeah, that's a really good point. Yeah, I guess anything, it sounds ideal when you have it like, okay, this is obviously, it makes sense, but it kind of eliminates all of the factors of, all the fixation that we do, any like, I guess, all the pre-processing or tissue preparation before you actually get to expand. Like, how much does that remove before you even get to that step, right?
Right.
Very true. Yeah, okay. So I guess, yeah, I'll look into that. I don't think so from what I've done with expansion so far, I haven't used SDS, but yeah, that's definitely something to keep in mind.
I mean, it doesn't affect it once the antibodies are on,
Mhm,
because once they're bound, and
mhm.
it's fine. But I mean, some antibodies aren't affected by it, some epitopes aren't affected by it, but others are. So I guess it's going to be antibody dependent. But that was the first thought in my head. So
Yeah, that's great. And yeah, someone also mentioned in the chat that antibody validation is definitely very critical.
Yeah.
For sure.
Has
Right.
anyone else seen that when they stain after expansion that the gel will get a haze to it? For
I
example,
personally never done it, so
if I stain with DAPI again after, the whole gel will have this light blue haze. You can still see the difference between the nuclei and the gel, but the whole gel gets a lot brighter.
I have overstained gels that I thought were light. And when I came back, there was definitely a background that was not there before, and I couldn't wash it out.
Yeah, so that when I was reading this paper, that really made me wonder, what the actual gel background looks like here.
They did mention several times that the background did not change significantly, but I didn't look carefully at the images and the quantification.
yeah, and I think they really only took images of so most of the figures that they actually show of the brain slice, that's not taken through confocal are they're under a black background and they're like adjusted to be white, and so you can't really see like you said, like a blue haze. So they didn't really show. Maybe it's in the supplemental, or they had quite a few pictures in the extended, but even then I didn't really see them take a picture of like what it looked like, the tissue after all this processing that they did. Okay. Anybody else want to comment on this figure? Or okay, so yeah, we have a comment in the chat. So Lauren said, we have never tried staining after expansion. We thought that maybe the antibodies would not penetrate as well. We're considering maybe doing 72-hour incubation to improve penetration, but as far as I can tell from skimming the paper, they only did overnight staining. And then Vita said, I'm wondering whether playing with the concentration and timing of the nuclear staining would help with the background issue.
Possibly.
Mm-hmm. And then Brian said, we also do exclusively staining followed by expansion. We haven't tried this approach. But yeah, same here. I've only done like staining before, and even then, Claire and I have talked about, what concentration should we be using? Do we just use what we normally do for like a 200 micron thick brain slice? And in the beginning, when I just started, before even expansion, just the staining process, we noticed that a lot of the staining molecules would kind of it wouldn't penetrate through to the middle of the brain slice, even though a lot of them seem to reside on the outer or the perimeters of the tissue. And so we thought, is that because of our staining protocol? Or it's weird because I drip the stain directly onto the brain slice. It's not like it's just on the perimeter or on the outside. So how come, is it just an artifact of the structure of the brain, and maybe there are just more cells and stuff on the outside? Or is a generally a like tissue penetration staining issue. There's another comment that said, one of our problems is signal dilution, and I'm thinking that staining after expansion could conceivably mitigate that, assuming the antigens aren't limiting. Yeah. All right.
Not sure. I do pancreas, and when I did the expansion on pancreas and I did the DAPI after the expansion, I did it for, I think, 2 hours. I did the DAPI staining after the expansion for 2 hours and then washed it out. And it had pretty much stained uniformly all the way through.
Okay.
So
I've even done 15 minutes and it seems fine. But I've noticed it works way better to stain
Yeah.
after for cells than for tissue.
And so, Mariana, when you said 15 minutes, that's for brain tissue or for just cells?
I've tried it on lung tissue and cells. And I think esophageal tissue as well.
Okay. So yeah, I guess I'll just read it out in the chat or the comments in the chat, just so the recording will have the whole conversation. But so Claire responded to Brian saying, yes, I think that it is the main thing that they're trying to overcome with this technique. Maybe we could still try reaching out to the authors to see if someone might come to one of our future meetings. That would be absolutely great. And Brian said, DAPI is pretty small compared to antibodies. So, yep, and I think there was one more comment or someone unmuted right before. No. Okay. So I guess we can move on to the other figure of the paper. And so here, this is figure 2 in the paper where they showed that they were validating this expansion revealing technique using synapses and comparison with the DNA paint. And so here at the top, you could see that, which was the image that I was talking about, like one of the few that they showed of the brain slice. So this is a wide field, a low mag wide field image of the mouse brain slice that they had stained with DAPI. And yeah, those dotted boxes up here show the somatosensory cortex that they used for future figures. And then the bottom here shows the B, C, and D shows the confocal images with maximum intensity projections of the representative fields of view. And so, yeah, they did immunostaining against antibody PSD95, Homer 1, and Bassoon, and Shank 3. So anyone have any comments on this one specifically?
Ellie, I found it a bit unclear. Are these just multiple examples of the expansion R, or are they showing other methods? Is it just multiple examples of different areas of the brain?
Yeah, I think it's just the different locations that they took.
But it's all the expansion R. It's not comparing it to other techniques in this figure.
In this specific figure, no. This one is just the expansion R.
Okay. And there's a question from Brian there for non-neurobiologists. What are these antigens? I don't know if anybody in this call knows the antigens well. I would assume they've picked things that are known to be separated within the synapse and the pre and post synaptic areas, but I don't know if anybody knows more. So Dita says Schenck and Homer are also associated with the postsynaptic density. Do you know if they're meant to be, in a certain order or in I guess they really, when we think about colocalization, nothing can really be colocalized and can't be in the exact same place. So. I think you could go on, Natalie. No, they tend to have a specific distribution. Okay, thank you.
Okay. So yeah, anyone else? Oh. Okay, that's good to know. Machinery. Okay, so Brian responded to that. Cool. So this is a postsynaptic pile of proteins, essentially. Vita said yes, but the PSD is complex and very interesting. And Chris also said that bassoon is a synaptic signaling machinery protein found right at the active zone. Okay, so it's presynaptic, the bassoon. All right. Anyone have any other comments on these figures or any concerns about like the distances or how they measured their data? Anything like that?
Hello, I'm Elizabeth from Mexico. And precisely that was one of my main doubts with the paper, because they say they achieve resolution about 20 nanometers, more or less 20 nanometers. And then in the paper, they also say that the biological distance between these proteins is 20 nanometer. So it was kind of confusing this part for me, because I don't know, I don't know how are these proteins distributed, and if the resolution that we achieve, considering the biological distances these proteins are supposed to have, give us enough information or reliable information about what we are getting. And I know that they did a lot of controls, and also that was something that I wanted to say, that as Brenda suggested, this is antibody dependent, so I think it's very important that when we are working with a new tissue or with a new protein or something, to have controls to know if what we are doing is working well. So I don't know if someone can comment a little bit about this stuff of the biological unit in relation with the resolution that they achieve.
I think one comment, because you can see in panel E that they go well below 20 nanometers. But here they're looking at the distance between two different molecules. So you can think of it kind of like STORM. If they can localize the center of the PSD95 stained spot versus the center of the Schenck 3 spot, because they're different colors, you can probably resolve those distances center to center a little bit lower than 20 nanometers. So that would be my guess for why they can actually go a bit below 20 nanometers on that panel E. Whereas if it was just looking between the two molecules with the same color, you wouldn't be able to resolve it.
Yep, and then Dita said in the chat box that the synaptic cleft is typically 20 to 40 nanometers. So that kind of gives them the rough ballpark of if they can see that these proteins are separated at the pre and post synaptic clefts, that they're within 20 to 40 nanometers, I assume. Does anyone else have any comments for this, or should we move on to the next? We're good to move on. Perfect.
They also, hi, this is Carrie. I'm at Vanderbilt as well. Hi, Brenda. they also said, I can't find it now in the paper exactly, but they're talking about how different antibodies will also join to different subunits of different proteins, which I found very interesting. So if you have a Homer 1 and from one particular manufacturer and then a Homer 1 from another manufacturer, what that would end up doing for your resolution.
Anybody have comments to follow up on that? Or?
Yes. Hello, this is George from St. Jude Children's Research Hospital. I know there was another expansion paper that used multiple different anti-tubulin antibodies to both increase signal and kind of create a more complete viewing of the microtubule. So that, yeah, that might be an interesting strategy moving forward, but it requires a lot of antibody testing, which is, you know, not most people's favorite experiment.
George, do you mind sharing that link?
Yeah, I'll look and see if I can find the paper.
Thank you.
And feel free to upload it to the publications form then. And then we can help you distribute that to everyone, even to those who aren't here today. Okay. All right. We're moving on.
There was just one more comment there, Natalie, from Dita about the size of the antibodies and that, you know, this resolution is similar to the actual size of the antibodies, and suggesting that looking at more nanobodies or camelids would be another way to get around this. And I don't know if anyone else would comment, but I know we tried doing that with some STED imaging, but then because your nanobody is so much smaller, you have fewer fluorophores, and then your signal to noise is lower. So you're actually unable to resolve as well because you don't have as much signal. So it's really interesting, these kind of trade-offs you get, right? It's a smaller molecule, more localized to the epitope, but now you don't have as much signal. So it's a bit of a puzzle, I guess, to find the perfect settings.
And Brian, on that same note, commented that he's wondering whether about the physical size of the immunoglobulins. The typical IgG is 15 nanometers on its long axis, so an indirect immunofluorescence complex could be itself about 30 nanometers in size. So how can we be getting biologically meaningful data at about 20 nanometer resolution using tools that are so large? So yeah, both pretty much on the same note, but yes, very good points. And so yeah, and then in the next figure here, next one, yeah, this is the DNA paint that they compared their expansion revealing technique to. And yeah, in G, H, I, and J, they show more quantified measurements of their error or their distances between the staining that they did. So I'm personally not too well versed in like DNA paint and like the super resolution part of it. Is that like, so they just kind of paint each DNA molecule and take that as for the super resolution imaging, and like, so they know where everything is in that way. And then that's how they compare. So I think it's also on F, in the image F, it's a pre-expansion. So yeah, they didn't expand anything, and then they compared it to after they expanded on the right. So I think it should be the same sample here. And it's also the same field of view of the cultured neurons after immunostaining against synapse and blood. So anyone have any particular concerns about this? For those of you who have done DNA paint, is this something is this a reasonable result to have? If nobody really has any comments on this one, I could also just move on to the next.
Yeah, I think the quantification they did in those figures is fairly complex and probably takes a bit of time to digest and look at the equations they used and understand.
Mhm. Yep, that's okay. We can kind of move on to the next one first and then kind of come back to this if anybody has any other thoughts. So the next one here, they wanted to validate how their expansion revealing enhancement and the effective resolution in synapses within the mouse cortex. So in this image A here, so in figure A, they kind of show this low magnification again of a wide field image of the mouse brain slice with the DAPI staining, and then they specified with this dotted box what area or field of view that they want to zoom in on for their confocal. And in pretty much in B and H are after they did their expansion. And then these are, so with the expansion they did, they immunostained with antibodies against CAB 2.1, which is a calcium channel PQ type. And then they also did RIM 1,2 and PSD 95 and SYNGAP as well. Anyone have any comments for this one?
So I think the main point here, Natalie, was just to look at another area of the brain, right?
Yeah.
Okay.
Well, and to look at the other, like the calcium, I guess, the other antibody, other proteins also. So yeah, we can move on then. So So yeah, this is kind of just by extension, the rest of that figure that essentially shows the sin gap, the Homer, shank 3, and bassoon again. And so in this figure, they wanted to show how the calcium channel distributions participate in transsynaptic nano architecture. So they show another low mag wide field image. And then here they kind of use the same calcium 2.1 PSD 95 and REM1.2, and then they merged again together. And this was in different, I guess, levels of the brain. So L1, L2, 3, and L4.
Did they notice any resolution changes depending on what level they were in?
Any resolution changes? I don't think they said. I think they quantified it in the next figure. So I'm gonna skip to that here. So here they quantified each one for, this is their autocorrelation analysis for each of the different layers. So it doesn't seem like they saw much difference between the layers.
Sorry, not a brain person here. When you say layers, are you talking about layers of cortex, or are you talking about layers in the stack of
Layers of
image?
the
Layers of the brain?
Yeah, layers of the brain.
Okay, thank you.
No worries.
Natalie, maybe you could just go back to the previous figure and show the L1.
Yep.
Yeah. And panel A there, maybe just review that.
Yep, so this is panel A where they take, this is the L1, the L2, 3 that they took, and the L3 or L4. And so Valeria had questions. She asked, using different antibodies for different epitopes against the same proteins is different from using directly polyclonal. Polyclonal should never be used. And then Brian said, one of the big conclusions here seems to be that their expansion revealing approach increases signal to noise as well as resolution, but their expansion revealing protocol seems to involve staining pre-expansion, imaging, expanding, then staining again, and imaging again. So do they control for the effect of tertiary antibody to primary, secondary, and tertiary staining without expansion?
To address Valeria's question, using different antibodies for different epitopes is versus polyclonal. Polyclonal, yes, can always be used. The question is, are you using known different epitopes versus polyclonals, which are, you don't know what the different epitopes are. Sometimes using different antibodies can give you different results. I have used 2 different polyclonals to get the same, to boost signal, because one polyclonal will have one set of epitopes and a different polyclonal will have a different set of epitopes, and they will not necessarily overlap. For whatever reason, they just don't. So if you use both polyclonals together at the same time, or you use one polyclonal and a monoclonal, they will not, they'll have complementary overlapping epitopes and give me a improved signal for whatever reason. I hope that answers your question.
And does anybody have a response to Brian's question on different controlling for the effect of different or tertiary antibodies? Any comments? So George has said that, so going back to where, when he mentioned the other paper, he said that expansion SMLM did not use multiple antibodies against the same protein, but they give examples of the linkage error introduced by expanding the position of the antibodies relative to the epitope when the gel expands. So I guess that kind of ties into Brian's question as well. Okay. Perfect. Anybody have any other comments on these figures or? Okay, so I will. Move forward here. So going back to where they quantified the distances and the autocorrelation analysis for these different protein distributions. I guess, yeah, according to the different areas, they all look very consistent. Is this what is expected for anyone who is more familiar with the brain? Or, you know, are these too perfect? So yeah, George has posted in the chat the link to the paper that he referred to earlier and an additional paper from Hamel and Bouchard, who have also written a book chapter on post-expansion staining to reduce this linkage error.
And sorry, I should have written post-gel staining.
Post-gel staining, okay. Thank you for the correction. Anybody have any questions about this figure, or are we good to move on to the next one maybe? Okay. We'll move on here. So in this next one, they look at how expansion revealing can reveal periodic nanoclusters of the specific protein peptide in the fornix of Alzheimer's model. So this is where they kind of applied it to the disease application and looking at the beta amyloids, I think, in Alzheimer mice. And so A here is a epifluorescence image showing a sagittal section of this FX fat mice brain with the fornix highlighted right here. And that's the area that they focus on. And then they move on to the confocal images again here. So this, they did immunolabeling against the A-beta-42 peptide with 2 different monoclonal antibodies, 6E10 and 12F4. So, and then they do the pre-expansion and post. Any comments on this one? I guess it's interesting to see that the pre, you don't see anything. I guess it's just all dark. But I guess that does make sense if it's according to their, I guess, hypothesis. And so I guess I can also move on to the next one, which is pretty much part of the same figure. I just couldn't fit it on the same page. So here's the 12F4, pretty much the same experiment, but with a different antibody. And so they kind of use these figures to show that their post-expansion revealing staining is able to reveal these like periodic nanostructures of the beta amyloid, whereas for pre-expansion staining, you can really only detect like large plaque centers.
Did they ever discuss what their concentrations were for their pre and post expansion stainings? Did they use the same concentration for their primaries and secondaries for both? Or did they adjust it for what they were doing? So.
I think that was mentioned near the end when they, like at the end of the paper where they talk a bit more about their methods. I think they did use the same from what I read, but I'll double check. Give me one second.
I was just thinking that if it wasn't strong enough for the pre-expansion, that would also give the results that they're showing right now. And that if they, you know, just, I mean, depending on what their different types of concentrations would be, could even give you these types of results.
Yeah, that's really true. I think they did keep it consistent for their staining protocols for both. But yeah, that's a really good point. So I guess we'll move on to the next set. Here they pretty much just focused pretty much more on the same model where they just show that they're able to review the co-localized clusters of this peptide with potassium and sodium ion channels instead of the proteins that they kind of targeted last time. And here, yeah, so they have pretty similar results in terms of Um, then, so this is their, here they're showing the expansion revealed confocal image, the maximum intensity projection showing the A beta 42 in magenta and the KV 7.2 clusters in yellow. And the top is indicated with cross-section profile, or this is the cross-section profile, yeah, there. And D is the histogram that shows distances between adjacent, the A beta 42 in magenta and the KV 7.2 in yellow. And this is all the distance along image segments of the axons. Any comments, any concerns? So I'll go through the other, there's only a few more left, so I can go through those and then we can kind of loop back and I'll let everybody kind of think about it, process it, or look at it on their own. So this here is the histogram and G showing the fraction of the A beta 42 clusters co-localizing with the KV 7.2 clusters. And then these are also along individual segments of axons. So they show that this is 50 clustered pairs from 9 different axonal segments taken from 2 mice. And then H shows a histogram showing the distance between the centroids of co-localized the same proteins A beta 42 and KV 7.2 clusters. And then I shows the diameters of these clusters. So I'm not too familiar with the distances between these 2 proteins, so I guess they just wanted to once again demonstrate that they can, it's reproducible within different, studying different proteins and such. And then here is the last figure that shows the analysis of their, the peptide and the potassium, the A beta 42 peptide and potassium ion channel nanoclusters and shapes and their relationships. So another violin plot here. They measured the length of the first principal axis and also the volume of the different puncta. Yeah, aside from, I guess, everything that we've already discussed in the earlier figures, or just the design of their original or the first few experiments, does anyone have any comments or like concerns that they think should be brought up in terms of the other experiments or the model that they used.
I think a fair number of the concerns came up along the way, right? With the whole aspect of antibody staining and the size of the antibodies.
Yeah,
And then I guess the fact we don't have many expert neurobiologists here is makes it a bit challenging, right?
yeah.
Natalie, do you know offhand the difference in the protocol that they've used to try to retain the proteins after expansion? Because they mentioned that they had another technique they called iterative expansion, but now they've called this retention because
Yeah.
they're retaining the proteins. Is there specific changes in the protocol that you're aware of that do that, or is it kind of in the details and not obvious?
It's more in the details. They do provide like they have like the protocol that they use, but looking here, it looks very similar to pretty much just iterative, but I think it's just the fact that they're trying to really anchor all of the proteins or most of the proteins to the first hydrogel before. Because that's one of the main differences that they had between this iterative and this new revealing technique.
So they're doing more cross-linking at the early steps.
Yeah.
Thank you.
I have a quick comment. In the paper, it says that these are like 50 to 100 micron thick sections. And then with the iterative expansion, it says it's like 15 to 20 times bigger. So how did they do turf that well if it's so thick? Like maybe I'm missing something. I'm wondering if anyone else had that idea.
You mean in their iterative one or this one? The iterative.
Mm-hmm.
Yeah, that I'm not too sure. I've never done turf, so I'm not too familiar with that. Oh, I think there's Valeria asked if you could please repeat the question.
Maybe I'm not reading it properly, but it says that it's a 15 to 20 times expansion factor. And the tissue is already 50 to 100 micron thick. And then they did turf on this sample. I'm just wondering, maybe I don't understand turf as well as I thought I did, but how did they image the entire sample through, or are they only just looking at that one part? And then if they are only looking at one part, why did they make sections so thick to begin with?
Which figure did they refer to turf in?
It is and it's on page 1069.
Unknown caller.
Is it in the?
It's in the DNA paint section. So do they only do it before expansion?
I think they would just only be looking at the surface, as I think Chris had mentioned in the chat as well. So they'd just be looking at whatever structures happen to be at the surface of the tissue.
Okay.
So Brian had also commented that they're presuming a lot of their validation on previously published immunogold or other data on synaptic architecture. And then he also mentioned that the issue of the size of immunoglobulins or the size of the, or WRT, the size of the structures isn't an issue because we're looking at the fluorescence after expansion. So the effective resolution can be smaller than the size of the immunoglobulin complexes because the epitopes have been moved away from each other by the expansion. Dita also had a question, a more general one. So she said that we know that aldehyde fixation substantially alters tissue relative to high pressure freezing. And in fact, that aldehyde fixation may disproportionately affect the presynaptic terminal, as shown in EM. How concerned ought we to be when using aldehyde fixation with optical nanoscopy as opposed to EM? So I think it was mentioned that like aldehyde fixation does cause a lot of like distortion. And so even in this paper, I think if not this one, but another one that I recently read, they kind of tried to reduce using aldehyde fixation for preparing the samples, especially when expanding, and then afterwards it becomes, you lose a lot of the structure apparently. But yeah, I've only read that to kind of avoid it. I don't really know too much about like to what extent it causes damage, I guess. Yeah, does anybody have any other experiences with this? Or any other questions?
So I'd like to thank you, Natalie, for taking the time to read through this in detail and present the paper. And I think we've brought up some good comments and pitfalls like fixation and antibody staining and so on that we can continue to explore at future meetings.
Yeah, no worries. And I guess on that note, I did have a question for you all about staining. And so I'm personally, Claire and I are interested in like actin staining. And I guess I previously worked a bit with this stain lectin 649 dialite that's used for blood vessels and like to stain endothelial cells, but I haven't had much luck retaining the stain after expansion. I was wondering if any of you have more experience with a blood vessel stain or a vasculature stain, and whether if you know this one is known to not work and I should kind of just try to think of another option, or maybe it's just my protocol.
It is not just your protocol. I've tried to use a tomato lectin in expansion microscopy, and I know that it stains really well before the expansion, but after the expansion, I've noticed that it kind of fades away a bit. And I don't know if that's because that it is stained with Alexaflor 647 and I've chewed up the staining because of the protease that I'm using, or if it's just floating off. This I have not determined yet. I do have an option for you. I have a very nice sheep CD31 that works really well.
Okay.
Let me send you an email.
Okay. That's great. Thank you so much. Hello.
That was really interesting. I appreciate all of the effort you've gone into preparing this, and I've got a non-useful solution to your vascular staining. You could work on zebrafish. There are lots of transgenics. Lots of transgenics with GFP in the vasculature in zebrafish. But I think the lectin problems are almost certainly that they're not fixing, right? Because they're interacting with carbohydrates. There's no aldehydes for them
Mhm.
no primary amines.
So they're just washing away.
They're just washing
Yeah.
away. Yeah.
Mm.
But so two things, just we work on zebrafish, and we do all of this with whole mounts or dissected bit eyeballs, so not sections. So are all of you doing your expansion on like tissue sections or tissue culture preparations?
Tissue blocks, yeah.
So nobody, yeah, because I think one of the hardest parts for us is the sample prep in the first place. And we're getting a lot of dilution of our signals after expansion, and that's one of the big things that I'd like to try to find solutions to. And this idea of staining after the expansion is probably a good one.
But the volume of the amount of antibody that you have to use to do that is,
Yeah.
yeah.
Mhm. Mhm.
The antibodies are expensive.
Yes, they are.
They've gotten a lot more expensive the last few years, for sure. Mhm.
Does anyone else find photo bleaching a real problem?
It's not been that big of a deal for me.
No.
No.
So do you do all of your expansions in the dark? We're like doing it all in a drawer in the lab because, yeah, they expand great, but there's not much signal left if we do it on the bench. We
Are you
able
sure
to
it's bleaching and not
just
just
dilution?
loss? Yeah, because
Yeah, well, it's definitely both.
Yeah.
Because certainly there's dilution, but we now are doing the expansion in the dark, and we're certainly able then to see the signals after expansion. But we
and so
initially
you do see it helps.
it
if you do it in the
definitely
dark?
helps. Yeah, and we're using this is using Alexa Fluor secondaries. So so we've got a 488 and a 555 are the two that we're looking at. But yeah,
Never.
photo bleaching and dilution are the two biggest problems.
Right.
Never had photo bleaching problems, and I just do mine out on my bench.
And then you're using the Dialight?
No, I use Alexa floors.
Oh, maybe you've just got a lot more signal to start with than we do.
That's entirely
Yours are
possible.
mostly GFP.
I
right?
do pancreas, so I have a lot of signal on my stuff too, so I
Yeah.
might just not be losing as much.
So for the expansion stuff, we're doing indirect immunofluorescence of epitopes.
Yeah.
And so we have Alexa 555 and Alexa 488 secondaries, and they are both definitely photo bleaching to some extent, but it's a combination. I think what happens is with the dilution, we wind up going right to the limits of our ability to detect, and then you lose another, even if it's only 15 or 20% to photo bleaching, it's gone. So
Yeah. The other
I've
thing
never
I
used that one.
The other thing I've wondered about is these are very aqueous environments, and the fluorophores are organic molecules, and whether there's also some quenching, because you can imagine if it's all packed in with other proteins, it's not a very aqueous environment in, say, unexpanded tissue. But when it's expanded, it's really sitting there free in an aqueous environment. So I don't know if there's anything you can do for that I don't know if anybody's even looked at that, but that's the first thing that I thought of. Just I come from a chemistry background, so
interesting idea.
yeah, and whether there might be something you can add to the buffer that might enhance the fluorescence.
Well, we have now started putting propylgalate in as Ross scavenger, but
I
I
was
had not
thinking,
thought about
yeah, more of
shifting
like
into a more organic.
viscosity or how hydrated it is. And I know it's a hydrated solution, and if you start putting something different, you might also get refractive index issues, but
Well, you might not
probably
shrink it.
something. Exactly. But probably something we're thinking about, whether there could be small molecules that could stabilize the fluorophores. Mhm.
Excellent. All
What
right.
about
Well, I
an
haven't.
isotols? I use an isotols in the 3D tissue imaging. I wonder if that would help stabilize it.
So
Mhm.
sugars,
Yeah,
is that primarily for osmolarity?
yeah. Well,
Okay.
I use that in for 3D tissue imaging, which is an entirely separate project, but I have to use that for refractive index matching for 3D tissue imaging.
Right.
Definitely worth thinking about. I'm not sure off the top of my head whether it would help. Yeah.
I'm not sure, but
Mhm.
I have
it was
another
random
meeting
thoughts.
I'm going to have to run. So
Yeah,
thank you again
thank you
very
f-
much.
Thank
Nice
you
to see
everybody
all.
for your contributions, and we'll the notes are available already in the document, and the RMS will put the video up if you want to review anything, or if you know anyone else who might be interested in listening to the topic we talked about today. So our next
Natalie,
meeting, February
I'll-
1st.
I'll send you an email.
Thank you.
Awesome. Thanks
All right,
very much.
thank you, everybody.
Yep.
Thanks, everyone.